Hey, my name is Madison. Like you said, I'm a post-bac scholar in the Lanyap program. And I'm going to tell you about a diversion on a typical trend that can be investigated in animals which naturally regenerate their appendages, and what these findings are going to mean for the future of regenerative medicine in human patients. So first I want to review the main motivation behind adapting natural regeneration mechanisms for treatment in human patients, mainly being the issue of medically necessary limb amputation that is disproportionately burdening the southern half of the United States. Two main causes of this is peripheral vascular disease, but also diabetes and traumatic injury. Sorry about those numbers, that's not correct. These two main causes are disproportionately affecting low-income communities in the United States and affecting third world countries and older populations globally. So these disease-related causes especially are only projected to increase by 2060, so it's becoming a more growing problem. Motivated by this problem, one goal of our research is to investigate the mechanisms behind natural appendage regeneration, specifically isolating elements that aren't present during homeostasis but then become activated after injury to mount a regenerative response. And that's why I want to tell you about a long-supported definition related to oxygen management and how our group has identified regeneration trends that challenge that long-standing definition. First, how we investigate oxygen management typically begins with globins. So I'm going to go quickly over a role of two major heme proteins which bind to oxygen, one being hemoglobin, which is involved in oxygen transport throughout the blood, the other being myoglobin, which is involved in traditionally oxygen storage exclusively within the muscle to prevent oxygen deprivation during periods of exercise or diving. And like I said, this is a traditional role, and I'll show you later how that is challenged. So natural regenerators have a variety of globins within their genome, specifically salamanders, very popular regeneration model. Axolotls are tetrapods that can regenerate their full appendages, organs, and even parts of their brain. Lungfishes are the closest fish to tetrapods, and Polypterus are the oldest member of the ray-finned fish clade. And both of these fishes utilize some conserved appendage regeneration mechanisms to axolotl and represent lineages which are useful to investigate how natural regeneration changed during the fin to limb transition. In addition, a teleos fish, this one is a zebrafish, only has one myoglobin gene duplicate, so it doesn't experience these excessive globin duplications, but zebrafish in particular have a variety of genetic editing resources available, which is why we want to utilize them in this project. So all of these animals have similar regeneration strategies, but divergent oxygen management solutions simultaneously. So for this project specifically, we're focusing on Polypterus and lungfish myoglobin genes because we have evidence that they're behaving non-traditionally. And it doesn't matter as much which globins these animals have, but it matters more which globins are expressed differentially between homeostasis and regeneration. So there's a few ways that we can analyze gene expression, but the most reliable one we found has been HCR fish. And just simply the methodology is to investigate gene expression patterns by binding fluorescent tags to an RNA target within the physical tissue at different regeneration stages. So therefore we can investigate target gene expression over time. And how we do that is we design a probe set that's designed to go into the tissue and bind to an RNA target of interest. In this case, it would be myoglobin. And then there's an amplification step where these things called hairpins have fluorescent markers attached to them. In this graph, fluorescence is represented by stars. And then there's an amplification step where hairpins go into the tissue, bind to the probe, and other hairpins are binding to hairpins until the amplification is over. And that amplification step is going to increase the fluorescence up to a point that matches the biological reality of the RNA expression. So before I show you the actual HCR fish images, I just want to orient us within the tissue. These are 2 polypterous fins, one at 0 days post-amputation, one at 3 days post-amputation. I'll be referring to that as DPA from now on. And these are just H&E stained fins. So the blue/purple stain in this image are acidic structures, cell nuclei, cartilage, and that line running through the middle is bone. And anything pink is cytoplasm, muscle, collagen. And that black square is going to mark the boundaries of the HCR image, which I'm going to show you in the next slide. So this is specifically muscle and skin that is not currently regenerating. So here it is. Here's the HCR with the beautiful fluorescence. So right here we see 2 myoglobin gene duplicates, MB1 in green and MB2 in red. MB1 seems to be exclusively expressed in the muscle cells, while MB2 takes on the role of being expressed in the endothelial cells, so the cells lining blood vessels that are traveling through the muscle and supplying oxygen. So also DAPI is the blue dots that you're seeing. Those are staining cell nuclei. So that makes this particular methodology really useful, because if we zoom in further than this magnification that you're seeing, you can get a sense of within the individual cell, where is the fluorescence popping up, whether that be in the nucleus of the cell or the cytoplasm. So how I would interpret this is myoglobin is behaving sort of traditionally here, but it is interesting to see this like specialization going on, where one is being expressed in the muscle cells and the other in the endothelial cells. That's not typical. So like I said, not very non-traditional, but still interesting. So now I'm going to show you a non-traditional behavior, specifically in a regenerating, so 3-DPA polypterous fin. And this is going to be a zoom in into this regenerating skin of the fin. So this is what we see. MB1 again is in green here, but it's being expressed in the skin, which is unexpected. Specifically, every skin layer with the exception of the basal epidermis, or the innermost layer of the skin, marked in red here. And like I said, myoglobin is behaving very non-traditionally. I'm wondering if these trends are reflecting oxygen management mechanisms during regeneration. Specifically, if myoglobin is specializing to deliver oxygen to the regenerating fin or the skin when vasculature is limited due to an injury having occurred in the area. So those are the big picture questions that categorizing the oxygen environment within the fin is going to help us answer. So now I want to show you just a snapshot of a lungfish fin at 0 and 3 DPA. So this is going to be the skin and the muscle in an area that is not regenerating, and I'll show you what the trends for myoglobin are in these tissues. So here it is. And 2 more myoglobin gene duplicates are notated in green and red here. MB3 seems to be specifically expressed in the muscle cells at both regeneration stages, while MB4 is doing something a little interesting. At 0 days post-amputation, MB4 is expressed in the muscle cells, but in every area that MB3 is not expressed. So there seems to be some sort of weird specialization even within the muscle cells going on that we didn't really expect. And then MB4 here at 3 DPA is expressed exclusively in the skin, while MB3 is sequestered to only the muscle, which is something that we especially didn't expect to happen. These really non-traditional myoglobin behaviors are maybe reflecting on trends that are happening within regeneration to deliver oxygen to tissues that need it. So those are like the big picture questions we need to answer with this. So now I want to show you some HCR images from a zebrafish tail fin. This is just a section of a zebrafish tail fin, and it's going to be at the very distal end. So this is what myoglobin is doing in a zebrafish tail fin, presumably. I just want to preface this by saying the zebrafish tail fin is made of mostly bony rays, so bones and skin. Notice how I did not say muscle. It's very strange to find myoglobin expressed in these cell types. And the circled green expression that you'll see there is partially, it could be expression in blood vessels running proximally distally, but there is work still ongoing to really figure out what is the actual biological reality of this tissue rather than just one instance of us seeing expression. We have work to do a whole mount HCR, which means we're going to do HCR on the entire zebrafish tail fin instead of just a section, so we can get a better sense of what's really going on with myoglobin. But this is an interesting preliminary picture that we have. So we also have gotten access to a zebrafish line that has no myoglobin whatsoever in their genome, and we want to do a functional assay to see if these fish can still regenerate even without having myoglobin at all. So that's another part of the project, to try to confirm the minimum functional requirements for regeneration related to oxygen management. So overall, investigating these non-traditional globin expression patterns is going to better inform us about the functional regeneration requirements and can help categorize the oxygen environment within the regenerating fin. And then this is one piece of a greater puzzle to unlock natural regeneration mechanisms in aquatic vertebrates in full, and then apply that knowledge to regenerative medicine in humans. I just want to acknowledge my mentors, my lab members, and especially the LingApp program being my source of funding. And if anybody needs really amazing fish pictures, I want to shout out my co-mentor. I used her pictures in my presentation today. She has an amazing photography company. So reach out to her if you'd like pictures for your publications or just for fun. And now I'm open to questions.
Yes, thanks for the nice talk. I thought that was very interesting, the differential expression patterns of myoglobin 3 versus 4 that you were showing. What is known about any functional differences between those two isoforms?
Right, so
oxygen binding properties, etc.
Right, so what you're asking about is something that I'm actually working on, but we still don't know for sure. So what I've been doing in this past week is generating the protein structure of those two isoforms and trying to analyze if there's any structural differences within the actual protein itself. And that work has been hit or miss, but that's the type of approaches that we're taking. So we're looking at actual protein structure, we're looking at the relationship of these isoforms on the physical chromosome, we're looking at gene synteny across species. We're doing a lot of comparative work to try to figure out any differences that could lead to functional changes like this.
Right, no, that sounds very useful. I was just thinking it might also be good to couple functional analyses of the proteins themselves. I think that's an absolutely crucial component.
I would love to do that. Unfortunately, lungfish have very little genomic resources available, specifically CRISPR-related ones that we could utilize in zebrafish. That's why we incorporate zebrafish, because they are much easier to do that type of work on. So with it, limited to this 1 year that I have, I unfortunately couldn't do any functional assays with those animals.